Welcome back, everybody. Welcome to this lecture on the cardiac cell membrane and how it regulates the ion influx and efflux. And I hope this really helps kind of tie everything together. We've talked a little bit about cardiac action potentials. We've talked a little bit about pacemaker potentials. We've talked about maybe you've seen some concepts on antiarrhythmics, but how does the cell actually handle this, and how do these mechanisms impact cellular function? And so here I've drawn you a schematic. We've got a cell here, we've got our main cell here, and then we've got our adjacent cell here, and these are cardiac myocyte cells. So these are our contractile cells. So we're going to talk about the function of our contractile cells. And so the first thing I want to talk about is, we've got this cell membrane in black, and the cell membrane is a phospholipid membrane that is not permeable to ions. So these ions that are coming in and out of the cell, they can't just pass through the cell. They have these specialized proteins that I've drawn to you here in either green, red, or blue. These specialized proteins that are ion channels. And these ion channels are what allow for the ions to flow in and out of the cell. The thing is with these ion channels, though, is that they're not always open, and the cell has mechanisms to control when they open and close and allow for ions to diffuse in and out of the cell. And so in order for these ions to even diffuse through these proteins, we have to have a gradient. We know that ions will only diffuse if there is a gradient, if it's going to go from a high gradient to a low gradient, or high concentration to a low concentration. And this cell here, the cardiac cell, it accomplishes this first and foremost with this protein that I drew right here in blue. And this is the sodium potassium ATPase. And the sodium potassium ATPase, what it does is it, as its name implies, it utilizes ATP, which is energy, and it pumps sodium from inside the cell to outside the cell. So we get a lot of sodium pumped out compared to what is in the cell. And then it takes this potassium from outside of the cell and it pumps it into the cell, so that we have more potassium in the cell than out of the cell. What it's doing is it's establishing a gradient of more sodium out and more potassium in the cell. And that's going to play a role in how we understand this action potential. And so ultimately, with all of the different ions, and we're not going to dive into it, what the cell is actually capable of doing is creating a membrane potential where there are more negative charges. I'm going to draw these negatives. There are more negative charges compared to positive charges in and out of the cell. So we see there's more positives on the outside and there's more negatives on the inside. And so that means that our resting membrane potential is negative. It's just the difference between the inside and the outside. And that is a negative. So if I come up here, I'm going to draw it on this kind of chart in the upper right corner. My resting membrane potential, this is my resting membrane potential. It is typically around negative 90 millivolts. That's the difference between the charges inside and outside of the cell. And so I'm sitting here in my resting membrane potential, and let's talk about what happens to fire off a cell, to activate one of these cells. And it all starts here with the adjacent cell. So this is the adjacent cell. And these adjacent cells are obviously, we think of action potentials are propagating from cell to cell to cell. They happen in a sequential manner. And so the activation of this cell, in fact, sends positively charged ions through these little gap junctions. We call these gap junctions. These are how our cells communicate. This is how our cell here in the middle knows when the previous cell has depolarized, because of these gap junctions. Gap junction. And so you get a little bit of positivity influx into the cell. And what that does is that makes this area a little bit more positive. Just right on this part of the cell is in fact a little bit more positive. And these green channels can sense the change, right? It can sense the change. And these are called voltage gated sodium channels. Voltage gated means that it senses that membrane potential where it's at. And so it senses that we're getting a little bit of an uptick in the difference between the inside and the outside. Notice how my slope here in the upper right hand corner was getting a little bit higher. And these voltage gated sodium channels, when it hits this threshold, which is my dotted line, the channels open up. And we said there's a lot of sodium outside the cell versus inside the cell. So if you open those channels, the sodium is going to rush in. And we're going to get sodium inside. Well, what happens when these positively charged sodiums come in? Well, it changes the area right next to it to become more positive, because these are positive ions. And that activates the next sodium channel. We get more sodium influx, which creates more positivity in the inside. And we send in the next set of sodium in. And then it does the same thing, and we get more sodium. And so we get all this sodium that is now coming into the cell, and it making everything more and more positive. And so that, if we look on the upper right hand corner of the video, that is where we get our phase 0 of depolarization. That is our big upstroke in our action potential. And at the same time, these potassiums that are inside, these channels start to open up a little bit. And so we start getting a little bit of potassium coming out. And at the same time, we've got voltage gated calcium channels. And voltage gated calcium channels sense this membrane potential change, and they send calcium inside the cell. And we remember calcium, this is ultimately the activator of the cell. So when you think about, well, what about this whole system is causing cardiac contractility to actual muscle contract? It's the calcium. So that's really the goal, is to get calcium in the cell. But these voltage-gated calcium channels need the voltage, the membrane potential to change so that calcium comes into the cell. And this part of the action potential is where we see the level off, where we see calcium coming in, potassium coming out, and they kind of equal out, and we get a plateau of our membrane potential. And then, once the voltage-gated calcium channels have completed their work and they're done sending calcium in, we continue to get more and more potassium coming out of the cell, which then is going to bring our positive ions back outside of the cell. And that's going to bring our membrane potential back more negative until it flattens back out. And as you've noticed, what happens when we propagate this signal of sodium rushing into the cell, well, we're also going to get a little bit of leak of that sodium through these gap junctions into the next cell. And that is what's going to allow for this cell to trigger to depolarize. So that is going to serve as the trigger for the next cell to depolarize. And once this entire cycle is completed, we now have sodium that is rushed inside the cell, potassium that is rushed outside of the cell, and we've got this sodium-potassium ATPase to get to work for a split second and pump all of the sodium back out to reestablish our ion gradients and pump this potassium back in to reestablish the ion gradients. And so that is how we get cardiac depolarization. Depolarization is this phase right here. Depolarization. And then we get our repolarization. So that's when I say that depolarization is accomplished from our sodium rushing in, and our repolarization is accomplished by our potassium rushing out of the cell. And so as I talk you through our antiarrhythmics, I want you to think about this concept. I also want you to think about if we block these voltage-gated sodium channels, we're going to be decreasing the speed of our action potential. And at the same time, if we block these potassium channels, we are going to be prolonging or blocking the amount of time it takes for repolarization to occur, essentially extending out the time it takes to depolarize and repolarize together, or the refractory period. So I want you to think about that. Those are the two channels that are most impacted in this diagram. Remember, our calcium channel blockers are a little bit different. They're not necessarily blocking the calcium that we're talking about in this video. So I hope this schematic helps you understand what is occurring at the cellular level in terms of activation and kind of resetting of the cells, so that you understand how these medications in the future and how these ions play a role into that function. So I hope this helps, and post any questions or comments that you have. Thanks.